Okay, well this is officially my first ever remote webinar, so I'm talking to a room with some people in it and also an unknown number of folk who are online. So I think it's actually quite appropriate given the topic of today's session, which is all around digitisation and really embracing the technology that we have available to us. So I'm very excited about this. My talk, as David introduced, is about digitisation in insect collections, but to begin with I actually wanted to take a much broader perspective than that and actually have a bit of a backwards look over the way technology has changed in our world in just the last 10 to 12 years. So the first slide here is looking at IT infrastructure in the world since 2001. So you can see in 2001 GBIF was established. GBIF is the Global Biodiversity Information Facility, and this is really the global repository and aggregator for biodiversity data from primarily museum collections, although its remit is now much broader than that. We also have the establishment of Wikipedia, which is now very commonly used by most people, and most folks know about it. Creative Commons licensing was established. In 2003 we saw the release of Skype, which was the first piece of software that enabled face-to-face interaction using the internet instead of line-based systems. 2004 was the launch of Facebook, and social networking really got its start with Facebook. 2005 we saw Twitter being launched, and also the creation of cloud computing, which really was the start of a new economy in using files and sharing files over the cloud-based network rather than using fixed systems. Believe it or not, iPhones have only been around since 2007. This was also the year in which the Atlas of Living Australia was launched, and this is Australia's biodiversity information facility, and we've actually got a speaker today who will talk a lot more about the ALA. It was also when the first GBIF data portal was launched, and then in 2007 we saw the launch of iPads. So we've only had iPads in our lives for 3 years, which I find very difficult to believe. Mine is basically surgically implanted, I think. And really we now live our lives connected, most of us, to these ultra-mobile devices. Now the reason why I think this is important is because natural history collections do have a very traditional science base to them, taxonomy and systematics primarily, although there's a range of other sciences that use natural history collections. But the world has changed, and it seems clear to me that like written word and libraries going through a revolution probably 20 years ago into the digital age, we're looking at the exact same situation with natural history collections. The physical specimens will always remain important, but we need to embrace the technology that we have and make these collections available online. I can see that there's probably 3 areas in which natural history collections will begin to embrace technology, and the first is around developing IT infrastructure. Natural history collections will become immersed in an IT framework, and this framework needs to be flexible and evolve with IT. And my previous slide showed you just how quickly that's happening in today's world. Having said that, it needs to be user-friendly and also well integrated into things like our ultra-mobile devices so that it's accessible to everyday folk. The second pillar, I think, is around creating a community. It's our community of users that give natural history collections their value. So without them, we don't have a purpose to have a natural history collection. And this community, as my first point says, clearly articulates the purpose and the mission with collections. And it also helps us to develop partnerships with those users and publishers and establish strategies around how natural history collections can be used more powerfully. The third pillar is around delivering the data, and I suppose this is where my talk today will focus most. Delivering data is around exposing the currency, and when I say currency I mean the up-to-datedness of the data. It also exposes some things that we find a little bit scary, the reliability or unreliability of the data, and the authority or unauthority of the data. And we generally lump these under fitness for use. However, exposing and delivering data also supports data management. In my talk I'm going to show a couple of examples around how digitisation is actually contributing to the management and use of our collections through things like annotations, which then feed back to a physical collection and can improve the data quality. Data delivery also allows us to flexibly connect different kinds of data together. So we're looking at a situation where it's now entirely possible to connect your specimens to tissue samples from those specimens, DNA sequences, and various types of media such as images and sounds and so on. And overall, what we want to do is make sure that our data delivery and use represents best practice at all times for all of our publishers and users so that we can maintain the integrity of the data. So really what I'm trying to say to you is that I see the future of natural history collections about taking the data that currently resides with physical specimens, beautiful physical specimens in a natural history collection, and turning them into web-based knowledge products that can be used by the community. And these are not just taxonomists and systematists. This includes our policy and government organisations, citizen scientists groups, environmental groups. There's a much broader range of users now. While also looping right back into contributing to best practice management of our collection. So therefore, positive feedback loop that actually improves the data that we're delivering. So let me bring this down now to something very specific, and this is because my personal experience is around managing a very large insect collection in Australia. It's the Australian National Insect Collection based in Canberra. It's under the auspices of CSIRO, and we have about 12 million specimens, so it's quite a large collection. Now, digitising an insect collection, and digitisation can involve databasing a specimen or imaging it, is very challenging for a number of reasons. So first of all, we do have large numbers of specimens. We have databased about 500,000 individual specimens in our collection. Now for a wildlife collection, that represents almost 100% of their entire holdings. For us, it's about 5%. And it's taken us 10 years to get there. So you can start to get a handle on the scale of the problem. Another issue is that identifications, authoritative identifications of specimens is difficult, uncertain, and sometimes unavailable. In Australia, we estimate that anywhere up to 80% of our invertebrate fauna is currently undescribed to science. So you can understand why putting names on things in our particular collection is very challenging. Furthermore, and this is more operational, but handling specimens for digitisation is always risky, and insect specimens are particularly fragile. The way that we manage our collections is that we pin a specimen and put the label information underneath. While this makes sure that the label information is always associated with the specimen, it makes it very hard to read that information unless you handle the specimen or pull the label off. Things like butterflies and large beetles are obviously particularly obscuring of label data. Now I've just got a couple of images on the screen here that sort of show the opposite side though, which is when you have very small specimens, you can see the label data, but you can't really see the specimen. You'll notice this is quite a magnified image. So there are a number of challenges. However, this hasn't deterred us from looking at the problem. This is some data that I've taken from the Atlas of Living Australia, and down the bottom you can see a list of the various collections that hold insect specimens. The dark blue bars represent how much of their entire collection, up to 100%, has been digitised. So you can see across the board, there's only a handful that have reached somewhere in the vicinity of half their collection having been digitised. And those ones are actually very small. The Australian National Insect Collection is the second one on the bar there, so you can see that our 500,000 records doesn't count for a hell of a lot in comparison to our entire collection. So it's a big problem. These are the biggest publishers of Australian insect data, again taken from the Atlas of Living Australia. So we have sort of four big players there: the Australian Museum, AM, the Australian National Insect Collection, ANIC, Museum Victoria, MV, and the Queensland Department of Primary Industries Collection. The others are much smaller contributors of data. So another issue is that even though some of us are publishing our data, it's skewed towards some collections or some parts of the country, and so we have to be mindful of the gaps that this might cause in the data. How much more do we think we have to go? This is just a back of the envelope calculation, but roughly in Australian collections we've digitised about 5%, so we have a long way to go. And clearly doing one specimen at a time is not necessarily the best approach to take. So we can look at a couple of large-scale or mass digitisation methods, and one that has certainly been adopted by a number of collections around the world is whole draw imaging. There was a recent ZooKeys special edition all about mass digitisation projects, so if you are interested in looking into this more, I'd encourage you to have a look at ZooKeys issue 209. Whole draw imaging is around taking an insect draw, putting it under a camera. It doesn't have to be a single lens reflex camera, it can also be a scanning system. Taking a high resolution image and then putting that up online. In Australia, a couple of museums are doing this fairly systematically. Queensland Museum is one. They're using a high resolution Hasselblad camera, and the image in the top there is taken from one of their draws. And then the collection I'm associated with, ANIC, is the second collection, and one of our images is there at the bottom. We're using a system called SatScan, which was originally designed for artwork, and it actually takes a series of scanned images across the whole draw and then stitches them together. The resulting image, when you see it online, is actually better than you would get with the naked eye. So you are actually getting a slightly magnified image. Another way in which we can get this information up online is to be focused. We can't do everything, so let's take priorities. One of the priorities is holotypes. They're nominally the most valuable specimens in our collection because they are the name-bearing type. Again, we have a few museums in Australia who are focusing on imaging their holotypes and getting these up on the Atlas of Living Australia. The example that I've got here is taken from the South Australian Museum project. They focused on making sure that all the cicada holotypes were imaged. They've provided a dorsal image, a lateral image, but most importantly, they've also photographed the labels. Now what this means is that anyone looking at this information or this record, if they don't have all the information they're after on the database record, they can look to the label image and acquire additional information. I'm going to touch on that again in a minute. This then actually formed part of a national project that was funded by the Atlas of Living Australia a couple of years ago to actually digitise all of Australia's cicada and bee type specimens. This project is almost complete and all of the data is currently being uploaded to the ALA. So for the first time, anyone interested in either of these groups will be able to access data and images for all the type specimens for all the Australian fauna that are held in Australian collections. Another challenging part of insect collection holdings are the bulk samples. These are samples that have usually been collected from indiscriminate sampling methods like malaise traps, and they're often mixed orders, anything from collembola and mites through to beetles and flies, depending on the method that was used. Sorting them out is very time consuming, and often they sit in drawers or jars and collections and they're virtually unused. The problem is that unless you have the time and the money to go and physically visit the collection and sort through the material, you don't know what's in the vial. We're suggesting that perhaps we should begin imaging these samples, so spreading them out on a tray, as you can see in the bottom image here, and including the label information, and then attaching that image to the record of the bulk sampling. And that record would include when and where the sampling took place. What this means is that you might not be able to look at these images and necessarily identify material to genus or species. In a lot of cases, you can identify to family. And as a researcher working on it's much more valuable than not knowing what's in the vial at all. It also unlocks a whole range of other information that we currently don't have access to, including things like diversity of orders at a particular site and a particular point in time. And we're also trying to use this process with a project CSIRO is running in the Pilbara region, where they're bulk sampling in an area that we really have no understanding of the diversity, and we're using destructive molecular methods to sample the molecular diversity. By taking these images, we've actually got a record of what we collected prior to destructive sampling. Now I just want to move a little bit away from the different types of methods that we're employing in insect collections and just touch on the fact that one of the challenges we face is a lack of resources to undertake this kind of work. The insect collection I work in is no different. We have a lack of resourcing both in staffing and funding, and it's very difficult to justify moving attention away and maintenance of the collection onto an activity that's focused on digitisation. This is where workflow is extraordinarily important. This is an example of the workflow we established for whole draw imaging. But the point is that at any one place in this workflow, we can value add. If we're going to get a draw out of the collection to check for taxonomy or pest damage or to top up the chemicals.
Welcome to GoToWebinar, Webinars Made Easy.
And I think possibly my colleague Bill Nelson, who's talking today, he may touch on this a little bit more. The other thing that I wanted to talk about is that acquiring the data, the metadata associated with the specimen, isn't an all or nothing exercise. We've already established that database of individual specimens is very time consuming. If we were to continue the process in ANIC, it's going to take us another 250 years to completely database all of our specimens. And that's without any accessioning of material at all. So we need to, I think, let that go. Instead, what we need to look at is, well, what information can we acquire by putting images up online and doing things like crowdsourcing? Our collection is involved in putting images of specimens and labels up on the Biodiversity Volunteer Portal through the ALA, and we're engaging online volunteers to do the databasing for us. There are still some challenges with this, and perhaps the most obvious one at the moment is that it's difficult to link that information back into our collection management system in a very easy way. There's no sort of click of a button and that data is sucked back into the CMS. We need to verify, then check it, and we also need to make sure that we haven't repeated information like collecting localities. But on the bright side, the data that has been entered by volunteers is already available for viewing on the ALA. It's not waiting for us to verify it. So you can access it now and see the distributions. And if you're not confident in the data, if you would prefer not to use it, then you can filter it out. But the point is, it's there, and I'm leaving the decision up to the user. I'm not making that decision for them. The other part of this is that it's not realistic to capture every piece of information. The couple of images here show that any one particular specimen can have a multitude of different pieces of information. We've got a slide of cockroach genitalia at the top. You can see what the species is. We can see that the sex is a male. We can see it's a type specimen. It was mounted in Uparel on a particular date in 1979. We don't necessarily need to spend the time entering all that information in. So perhaps prioritising it to the identification, the locality, the collector and the date, and then supplying an image of the label puts the power in the user's hands to extract any additional information if and when they need it. So just finally, I'd like to discuss a little bit about my experience with utilising these sorts of methodologies for managing my collection. This was an example of a data point that was up on the ALA and had been geolocated to a point in Central Australia. And it's for a species of dragonfly called the Mount Lewis blue streak. Mount Lewis is located in the wet tropics on the tropical North Queensland coast. So if you have a look at the bottom there, you can see that an issue was flagged by one of our community of users. I don't know Rhino Richter personally. And he has commented that this species is only known from Mount Lewis. Using this information, I was able to go back into the collection and confirm that yes, this geolocation was wrong in our database, and I fixed it. And you can see here, there's a photograph of the original specimen. It's just a papered specimen. It hasn't been pinned or mounted. The locality information was very sparse. Mount Lewis 3As is the only information we had there. But in our collection management system, we're able to confirm, and using the fact that it's a published species, we could confirm the locality and fix up the record. I then went into the Atlas of Living Australia and replied to Rhino Richter stating, thank you very much, your comment was correct and I've now fixed the record. So here's a beautiful example of how we can use digitisation to improve our own data quality. So in summary, I'd just like to list a few reasons why digitising has a number of benefits for collections. It provides virtual access to specimens and data, and this is an increasing problem in a world where funding is limited. And we also want to limit our carbon footprint. It can encourage remote curation of unsorted specimens and encourage remote curation of data, as I've shown you. It delivers the specimen metadata. It provides a method for auditing collections, and particularly large, complicated ones like insect collections. It contributes to improving collection management practices and also operational things like loans. I can show an image now of our collection to a potential borrower and he or she can tell me exactly what they want or if they want anything at all. It permits morphometric analysis of specimens and in fact there's a paper about this in Zuki's issue 276 I think about using images of dragonfly wings and demonstrating that it's actually no more or less accurate than hand measuring with calipers. promotes community engagement and improves our data quality and our fitness for use. I would just like to acknowledge that list of people and organisations and teams for their support, their ideas and involvement, and thank you very much for your attendance. Are there any questions? No, nothing from the, oh, one from the back? Yes.
Yeah, in your, so when you're using the crowdsourcing, you say you need to verify that. You verify the volunteer, or you do that individually by assessing their quality rather than each individual record, which is another way of being split by the group.
Yeah, there's three ways of going. Oh, I just need to repeat the question. So the question was, when we verify the data that's entered through crowdsourcing, do we verify the individual who entered the data, or do we verify each individual record as it's entered? There's really three ways. The first is that we can verify each individual record, but that gets very time consuming. The second is that we can vet some of our online volunteers and promote them into a role where they verify records on our behalf, and we're certainly doing that. The third way that we're not using with the VVP, but other online crowdsourcing portals are using, multiple entries. So they'll allow a particular record to be entered, say, up to 3 times, and where there's agreement, they'll accept that as a verified record. Yeah, thank you. Yes?
How many plans to link this because a lot of cases see a lot of stuff in the image, but we don't have to see the image description.
Yeah.
So copyright is left and
yeah. So just repeating the question for online, the question was, are we linking our specimen record data with the published descriptions of the species? I think probably John LaSalle may talk about this. He's the director of the ALA in one of the following talks. But we do link BHL, Biodiversity Heritage Library, records of species descriptions where they're available on species pages in the Atlas of Living Australia. So if you were to look at the species page for the Mount Lewis Blue Streak, providing that, as you say, the copyright has lapsed, it will provide you with a link to the publication where that species was described, all in one location.
So
to an electronic resource, yeah, to a PDF. Yeah. Okay.